exhibiting your PPTs with us.
Okay, where is that option? Participant share screen. Okay, this is. So are you? So now visible to you? Hello.
No, it's
No,
not.
Dr. Imran. Not visible.
Not visible. I'm not as much familiar.
Okay,
So,
Dr. Imran, let me set your PPTs here and just let me know which slide you are going to present. Just give me a moment.
I'm just orally presenting, and now I will come to the objectives. Now I'm
Okay.
just briefing you about the soybean and maize crops importance.
Okay.
Okay.
So
So
we
you may
are,
share with me the screen. I will present from there, please.
yes, just give me your
Yes.
slide number which you are
Yes.
presenting.
Okay. So this is my objectives. This is my objective that why we conducted this research. Our objectives was to investigate the impact of organic sources on crop productivity of soybean and maize. Please, the first one, please. Please, the first one. The previous slide, please. Hello, please. Show me the previous slide, please. No, the first one. The first one. Are you hearing me?
Yes, just give me the slide number. It's third, fourth, fifth. Which slide number you want to just go for the presentation?
He's presenting objectives, Mariam.
Okay. So
Previous one.
yes.
This one?
Yes, this one,
Okay.
yes. So why we conducted this research? So our main concern was to enhance the soybean and maize quality, also the quantity, but our focus was on quality. That's why we used organic sources and beneficial microbes. The next objective was to investigate the impact of beneficial microbes. We used two different microbes. One was fungi and the other was bacteria. In fungi, we used trichodermal fungi, and in bacteria, we used phosphate solubilizing bacteria. So we should need to know the impact of that too. The third objective was to investigate the impact of phosphorus level. We used three different levels of phosphorus: 50 kg, 75 kg, and 100 kg to know the impact of that phosphorus on the current crop and also on the residual effect on the previous crop. We also need to, the next objective was to study the response of maize and soybean to different interactions like organic sources, which organic sources will be better, which beneficial microbe will be better, and which organic sources with which phosphorus level was effective. Next, please. Next, please. Yes, this was the material method. We used experimental design, randomized complete blood design, and we conducted this experiment at Agriculture Research Institute, Mingawaraswat, which is well-known and prestigious organization among the research institutes. We used two different cropping systems. One was cereal based cropping system and the other was oil seed based cropping system. In cereal based, like maize wheat is cereal, while soybean and wheat was oil seed based cropping system. And we conducted this experiment twice in 2016-17 and 17-18. We harvested the crop on the 4th and the 2nd July, sown and harvested in October and November. Next, please. I would like to brief you what type of organic sources we used. In Swat, Pakistan, peach is known as hub of the peach crops. So here in Swat, peach lashes are also huge in a very huge quantity. So that's why we think that why we should not to reuse that waste material to minimize land pollution, also environmental pollution, and we use that and recycle the nutrients. So we collected that pitch residues and make we applied in fresh residues form, we make a compost from that, and we make a biochar from that. So what material were used? We just used only peach residues. There was peach leaves, peach dropped or fallen fruits having no stones and not decomposed. That was peach residues. In compost form, we applied that leaves, fruits having no stones, while well decomposed compost we used for application. In biochar preparation, here in fall season or in winter season, we making pruning. So we also used peach stem for biochar preparation, peach stone, its leaves and twigs, residual twigs which we cut in pruning season. Next, please. Next, please. Yes. So brief you that all economic parameters and all soil parameters. I will cover the economic analysis that which one cropping system was more profitable for the farmers and for the farming community of Pakistan. So on the basis of economic analysis, if we look peach residues, when we calculated the grain value, the maize grain value in control plots and in rest plots. In control plots, the maize grain value was very little, that was 78,350 in Pakistani rupees, while its VCR was almost neglected because we haven't applied any external residues that was controlled. When we compare the peach residues VCR value cost ratio, that was also little, and that was 4.49. You must all know that what is VCR. VCR mean when we expend 1 rupee, 1 Pakistani rupee, so it gives us in back 4.49 rupees. So the most profitable VCR showed by peach compost, which was 5.31. So I will come to the combined the 2 year combined economic analysis. So peach biochar showed more profitable effect in terms of the other residues. That was 4.85. Next, please. Next, please. Yes. Here we will compare which beneficial microbes was more profitable, phosphate solubilizing bacteria or trichoderma. So trichoderma showed every year very dramatic effect, which was the most significant among all the experiment in all cropping system, also in soybean wheat cropping system, and also in maize wheat cropping system. So trichoderma was the most amazing, and it gives us 12.14 VCR in first year and 16.20 in second year, and we combine these two, so it gives us as a whole 14.17 VCR ratio, which was the most amazing and most profitable and will be for the farmer and have been recommended. Next, please. This is by the soybean. In soybean, which residues either compost or biochar produced. So in first year, biochar produced more profitable yield and give us highest VCR, that was 6.44 in first year, while in second year peach compost gives us highest VCR, that was 3.5. But combine over the 2 year, when we conclude, it showed that peach compost gives us 5.48 VCR, while peach biochar gives us. So statistically, they both were similar. So in soybean wheat cropping system, we can recommend to the farmer that you may apply peach compost or peach biochar under the soybean wheat cropping system. Next, please. This was the rate of the soybean grain in 2016-17. Now it is too much high. So according to that, the value will be nowadays the value will be much more. Next, please. This is the VCR or the economic analysis for the phosphorus. You know that 50 kg phosphorus gives us little VCR, but moderate phosphorus level, that was 75 kg per hectare, it gives us highest value. The highest value cost ratio was 8.3 with 75 kg phosphorus per hectare. If we apply more phosphorus like 100 kg phosphorus per hectare, so our production cost will be increases. As our production cost increases, the VCR will be dropping. So that's why moderately level of phosphorus gives us highest VCR, and we have been recommended to the farmer that you must apply moderate level of phosphorus like 75 kg. So in first year, 75 kg gives us highest VCR. Also in second year, and when combined over the both years, so it also gives us again highest value, that was 8.2 under the soybean wheat cropping system. Next, please. Here is again economic analysis of the beneficial microbes, phosphate solubilizing, and bacteria and trichoderma. So trichoderma was amazing among all the cropping system, and here in soybean wheat cropping system, it gives us 14.31 VCR in first year, 14.39 in second year, and when combined over the both years, it gives us 14.35 VCR ratio, which was much more than the phosphate solubilizing bacteria. Next, please. So this was our conclusion, that we have concluded that growth parameter, dry matter partitioning in soybean wheat cropping system and also in maize wheat cropping system, its accumulation, yield components, soybean and maize grain yield, its phosphorus use efficiency, agronomic efficiency, partial pectoral productivity, soil carbon, phosphorus uptake, phosphorus content in plant cells, and so I just given you briefing on economic. So economics was also tremendously increased with integrated use of biochar with trichoderma and 100 kg 75 kg under both cropping system. This was the 100 kg was for the agronomic parameter and for the efficiency, but economically 75 kg was better than 100 kg. The grain yield of the succeeding wheat was higher than soybean was grown as preceding crop, while biochar, trichoderma, and phosphorus 100 kg was better for the cumulative profitability and income enhancement under soybean wheat and maize wheat cropping system. Next, please. Yes, conclusively, grain yield of soybean and maize was higher with biochar, soil incorporation of trichoderma. So we have applied trichoderma as a soil incorporation, and stabilizing was mixed with the seed. So soil incorporation of trichoderma was better than mixing phosphate solubilizing bacteria with seed. It was much better than that. So and phosphorus at the rate of 100 kg can improve soybean and maize as well as subsequent wheat yield. So 100 kg here. That's why here give us the subsequent yield or succeeding wheat yield. So when you apply phosphorus, so it is absorbed with soil and it available in next season. So when you apply more phosphorus in this season, so more phosphorus will be available in coming season. So that's why 100 kg showed better result as compared to the 75 and 50 kg. Next, please. This was our recommendation that Massive increase in soybean and maize production was noted with integrated use of biochar plus trichoderma and 100 kg phosphorus under both cropping system. On the basis of cumulative economic evaluation, application of biochar and trichoderma with 100 kg phosphorus per hectare under maize-wheat and soybean-wheat system should be recommended for the growers under the northern climatic scenario of Pakistan. And from the sustainability point of view, soybean-wheat system enhanced soil organic carbon and phosphorus content in soil as well as subsequent wheat yield, and will be recommended to improve soil structure health under the application of biochar, trichoderma, and phosphorus at the rate of 100 kg per hectare. Next, please. The result from the second year soybean wheat and maize wheat cropping system concluded that the integration of biochar with trichoderma and phosphorus at the rate of 100 kg could improve soybean and maize as well as subsequent wheat productivity under soil quality under different field condition. A positive relationship between soil phosphorus, soil organic carbon, and phosphorus use efficiency, grain yield, and harvest index suggested that peach biochar improve plant phosphorus uptake. Our result also indicated that potential of biochar to increase crop productivity and soil health for sustainable nutrients management by small landholding farmers communities in Pakistan. Next, please. I think it's finished, and many thanks for you listening.
Okay, thank you very much, Mr. Imran. So, do we have any question for Dr. Imran?
Yes, please.
Yeah, Dr. Imran, thank you for your nice presentation. My question is regarding your experimental design. You are saying that you are using a randomized complete block design. Actually,
Yes.
RCBD is quite common in agricultural field due to its ease of use. So, but the common problem in RCBD is the sources of variation. And to minimize the variations, normally you are using blocking. So, just I wanted to know that what type of blocking you have used in your design.
We have used for blocking, we have used just replication, for replication, that was our block. So not we did not use any split plot or any other arrangement. We just used we just increased our replication, and that was our blocking, and through this way we minimized our error, experimental error.
Okay, it's fine. Then the replications is increasing. Replications means initially you might have tried some replication designs, and after getting some variations, you might be increase the number of replications.
So we have also calculated collected climatic data too, that were on the solar radiation, wind humidity, wind speed, etc. But here they have been limited me up to 12 slides. That's why we not edit any specific pictures and other things in these slides. So we were limited to the 12 slides. That's why I not presented.
Yes, Dr. Imran, I do understand. Just I wanted to know the blocking you have taken. So now I will. Thank you for your answer.
Okay, thank you very much.
Thank you, yes.
I think we have another question. Yes, please.
Yes.
Try to be brief in questioning. Yes. This is another question from
Esther, can I go ahead?
Yes, please.
Thank you for the good presentation. I saw that you worked on phosphorus, and we know phosphorus is very important in legumes. And also we know that before you recommend the amount of phosphorus, it's also good to check on other nutrients which work with phosphorus, like molybdenum, boron, and the other micronutrients which normally help in biological nitrogen fixation. When you are setting the experiment, did you apply all the other deficient micronutrients as a flat rate, or you just worked on application of phosphorus without checking on the effects which might be caused by the other associated nutrients? Thank you.
Yes, thank you for your question. Yes, before application, before starting up the experiment, first we conducted we tested the soil that either our soil is phosphorus deficient or not. We just conducted our experiment on phosphorus deficient soil, but there were micronutrients were in insufficient quantity. So that's why we applied 70, 50, and 100 kg phosphorus. We were went for that soil which were phosphorus deficient. When there is enough phosphorus, so then no, there is no need of conducting of this type of experiment. So we must go for that side, that we tested the soil, that the soil must be phosphorus deficient. After that we applied that amount of phosphorus and beneficial microbes to know exact the impact of phosphorus. If we apply phosphorus to the adequate level of phosphorus, and we also apply beneficial microbes, so here in soil, in Swat soil or in Pakistani soil, here is a lot of rock phosphate. Here is also available phosphorus and unavailable phosphorus in huge quantity, but in unavailable condition. So when you also apply beneficial microbes, so that go per their side and they solubilize that. So that's why we first went to test the soil that is phosphorus deficient, and after that we conducted that.
Okay, thank you very much, Dr. Imran. So now we have
Thank
our
you so much.
next presenter. This is Mr. Santosh Kumar Karn from Sardar Bhagwan Singh University, India. So, Dr. Santosh, are you there? Yes. Can you please share your PPT? Are you not able to share?